Hmm. We need to project it. We didn't do that.
Oops.
I forgot. Here, actually, if you want me to do the slide so our zoom's not in the corner, I can do that. I don't need audio to do this part.
Get myself.
We have most of the TVs working.
I'll need to reference my notes.
Hey guys, welcome to the exam one review. How are you guys all feeling about this? It's like, I don't know, thumbs up, thumbs middle, thumbs down.
Cool.
Okay, well, hopefully this helps with that. Um, yeah, uh, cool. So today we'll be going over the four topics that are going to be on the exam. Protein structure and function, protein misfolding and regulation, protein trafficking and regulation, and gene expression regulation. Um, all the questions on that worksheet I sent out to you guys were going to be in like the middle of these slides, so we'll go through those, give you a few minutes to work on them, and then go over the answers. And they should be pretty similar to the type of stuff you'll see on the exam. I mean, you've all seen Laura's questions by now. You've seen like, I don't know, 50 problem set questions and quiz questions and all that stuff. So it's not too different from that. I'd say it's just a lot of them at the same time with the pressure of exam, which is not fun, but yeah. Okay.
Hi.
Ah.
Sorry. Okay. So yes, we have slides, but I like to do things a little differently. So the slides will be projected on the screen, but I'm gonna have you guys explain what's on the slide to us, to me. Is that okay?
Yeah.
All right.
However you want to.
So you'll have a mic and you're gonna tell me what you see on the slide. The best way, in my opinion, the best way to learn this content and to keep it in mind is to actually teach it to others or to yourself. If you can explain this out loud, then you'll do great on the exam. Is that okay with everyone? Well, I'm not giving you a choice, so. Alrighty, so the first one is amino acid structure and peptide bonds. What do we know? Who wants to start? What do we know about amino acid structures? Anything? Okay. What are amino acids made up of? On the slides, I'm not tricking you, I promise. No one? Come on, you can't all be shy.
Um, so there's like four groups. There's the carboxyl, the side chains, and the side group.
Of these four groups, which one does the hydrogen bond in? Does someone want to answer that? Of these side groups, where does the hydrogen bond comes from? Do we agree with that? Also with the carboxyl. Perfect. Alrighty. And what about peptide bond formation? How are the peptide bonds formed? Okay, that comment is on the screen. On the screen. A dehydration reaction. What does that mean? It makes water as the byproduct. Okay, but what is binding? Well, I guess you don't need to know this part about how it's made, do they?
Um,
Do you think?
that would sound.
No. Alrighty, cool.
This is
So,
where it is.
so amino acid structure, the amino acids are made up of four parts, the CH body, quote unquote body. We have the amino group, the carboxyl group, and the the R chain, the R. Wow, okay. The side chain. Now, this side chain, as you guys know, can be any of the 20 amino acids that we know. It could be non-polar, polar, polar charged, polar uncharged. And this often affects the type of interactions it can make with other polypeptide groups. And peptide bond formations are dehydration reaction. So we would lose that OH group on the carboxyl group, and that would form water with the H group on the amino group on another amino acid, and thus the peptide bond is formed, and now we have two chains of polypeptides of amino acids. And breaking a peptide bond is a hydrolysis reaction, and it needs water. Questions on this slide? Okay. Now on the polypeptide chains. What is the point? What is the protein backbone? What is it for? Why is it there?
It holds the protein together.
It holds the protein together. What molecules on the peptide is making this peptide? Is making this part of the backbone? Sorry, was that clear? Like what parts of the peptide are involved in the backbone formation? It's a one quiz one, guys. You don't remember? The O and C's. C's and the H's. No.
NCC.
Huh?
To make the backbone is NCC, NCC.
Yeah. Yeah. Okay. It holds the protein together. It is shared by all amino acids in the protein, and R groups on polypeptide chains face against each other in space. I don't think you need to know why, but can anyone guess why? It's fine. Okay. The carbon of one amino acid and the amino from a peptide bond. That was what we talked about with hydrolysis. Oh crap.
It's fine. I'll just keep it up.
Okay. That's what we talked about with the hydrolysis reaction, where you see the OH group of the carbonyl carbon, and the H of the nitrogen. I can't explain this in a way that's not chemistry heavy. Let's just say they form water at the end, right? The C loses its OH and the N loses its hydrogen, and that forms water, and then that causes the C and the N to bond. That's the most simplified way I can explain it without chemistry. All right. What are the four types of protein structure? And can you tell me about them? Guys, it's on the board.
The primary structure is just like the order that the amino acids are in, and it's usually not like affected by denaturing. Yeah.
Cool. Does anyone want the secondary structure?
It's the backbone interactions that make the beta sheets and alpha helices.
All right. What about tertiary structure?
Tertiary structure is based on like the folding of the like interaction between the R groups and how they interact.
What about quaternary structure? Quaternary structure, anyone?
It's when multiple subunits are interacting together. So multiple different polypeptides.
You just answered my next question. Cool. Do we see subunits and domains in tertiary structure? And why? Well, maybe not why. Do we see domains and
So we would only see domains, we would not see subunits because some subunits are only a part of quaternary structure.
was that?
Well, yes, yeah.
All right, now it's protein modeling structure. Did we have any questions about primary, secondary, tertiary, or quaternary structure? Okay, so we only have two on this slide, but can you guys tell me characteristics of ball and stick and backbone? When would you use ball and stick to show your protein of choice?
The problems they can show, the radius of atoms and cells, the covalent bonds between ones, and so it shows the backbone and the arc.
Okay, and when will you use the backbone model? Guys, this is very important. When will you use the backbone model to represent your protein? On the board. It's not a trick question, I promise. Okay, well, you use it to show the path of the polypeptide backbone. And it's a skeleton outline of the protein. No R groups are present there. And the next ones are ribbon and space fill. So when would you use ribbon? Or in what cases would you use ribbon structure?
To show the backbone and secondary structure.
Okay, then when will you use space fill?
See the overall shape of the protein.
Okay, cool. All right, and this is also our first question. It's also on the worksheet, as Keller has said before. So if a polypeptide chain is 400 amino acids long, and if an amino acid is the 81st amino acid in the chain, is it closer to the N terminus or the C terminus? If you want to see the alpha helices of this protein, but also observe R group interactions of residue 81 with amino acid 96, what model should we use? Give them time to solve it first, right? All right, give you about 2 minutes to answer this question on your own, then we'll come together and talk about it together.
Talk to people next to you.
Yeah.
Do that.
That's we like download the slides.
This worked a lot better during the mini midterm review.
Yeah.
We had a lot more interactions. I'll just look at that. I mean, I guess they're nervous. It's their first big exam, so I guess it's fair. It's very fair. How do you usually do it? Do you just read off the slides?
I just like, yeah, read off the slides. I take questions, based on how confused people are. I'll go into maybe more depth off the slides and like try and give analogies for stuff like that. And then I try to make the interactive part like the question part. Thanks. I might try that for the next one, just so it goes a little quicker too. 'Cause
Yeah,
it
yeah,
can
yeah.
take a long time if they,
Are
you
not
know,
answering.
answering. Yeah. Okay, that's probably good. It's not that long of questions. We will get longer ones. But yeah, so if we have the 81st amino acid in the chain, is it closer to the N terminus or the C terminus? You just raise your hand and shout it out. I heard someone say N terminus. Yes, that's correct. Good job. And then, so if we want to see the alpha helices of this protein, but also observe the R group interactions of that residue, which is another term for amino acid, what models should we use? Yeah, so you would use ribbon to see the alpha helices, but could you see those R groups interact with the ribbon? Exactly, yeah, you could use wireframe or ball and stick to overlay at those specific amino acids. Yeah, so there's the answer for the what I just said. So yeah. Cool. You want to take over? Oh,
Sure.
And then 3 and 4.
Right. Protein misfolding and regulation. So, sorry.
Okay.
Environmental factors that can affect protein folding. Anyone?
Any takers? Thermal energy, light energy, pH and salt, and the tertiary.
All right, so the way these environmental factors affect the protein may vary. So thermal energy, what do you guys think thermal energy will do to the protein if exposed to too much of it?
It makes it too hot, so the bonds would break and then the protein would break apart you in this fold.
Perfect. What about light energy? Light energy is pretty much the same as thermal energy. If it's too much, it'll radiate, make heat, etc. So what about pH and salt? These also have similar.
They're like the charges and stuff, so the bonds break.
Okay, cool. So denaturization is the process of the protein misfolding, and the proteins can renature if they're not aggregated, and this is through the help of chaperones. So chaperones help the protein refold. They help the protein refold. And if a protein is so when the protein is exposed, if a protein unfolds, its hydrophobic core is exposed to the polar environment, and that causes clumping. And excessive clumping leads to aggregation, and after aggregation, the protein cannot be saved. It'll be sent to the protease to be recycled.
Skip this one. We just said that too.
Yep.
Okay.
Okay. Another question. If you were to add salts to a solution of proteins, what levels of structure in the proteins would be affected and why? Maybe take a minute or so. Please do talk to the people around you.
How many slides do we have in this section? I'm just wondering how many slides we have in this section. Okay. Function mechanism, doing ATMs. Yeah.
Do you think that was a minute?
Yeah, that's probably good.
All right. Just shout it out. If you were to add salts to a solution of proteins, what levels of protein folding would be affected and why? Just shout it out. Guys, shout it out louder. Secondary?
Tertiary.
Tertiary? Okay. Any other takers? Do we agree with secondary and tertiary? Okay, that'd be something. So mainly tertiary and quaternary structures would be affected. Secondary structure would be affected, but not by a lot. So again, the salts affect the charged atoms, and these attract or repel the charges. And tertiary and quaternary are where charge R groups interactions happen, and secondary and hydrogen bonding is which happens during partial charges. Okay, protein function and mechanism. Ligands and substrates, and that is just anything that can bind to a protein or an enzyme. So, and protein ligand affinity is whether or not there's a good fit, quote unquote good fit, right? So, is there a question on Katie?
Later.
Okay. So molecules move around and bump into each other. So this is very, very random. So there is no, hey, I am a ligand and I need to bind to this protein, so I'm going to walk this specific path to get to this protein. It is very random. and it's in their environment. So the binding happens through random diffusion. So if they just randomly diffuse into the pocket of the binding pocket of their target protein, if they fit, they'll have high affinity. And if not, then they don't. And the dissociation constant measures how likely that ligand will dissociate. Regulating protein function, allostery. So protein function is regulated by changes in protein structure. Proteins adopt two or more conformational states and shift from one conformation to another. And dimerization is two things coming together to form one unit, right? And these changes in protein shape also change the function. Does this make sense? Do we have questions? Do you have anything to add?
Yeah. I would just add that like a good example of allostery is I don't know, a ligand binding a receptor or something. Like you have something floating around the cell and it binds a protein or a receptor, and then the protein will change its function. Actually, the receptor is not a good example. Let's just say if, an allosteric site is something besides the active site that can change the conformation of a protein. So if some regulator binds a protein, it can shift the conformation of that protein to alter its function. And that's what allostery is. It's something else binding at a place other than the active site to modify that protein or enzyme or whatever it is. Yeah.
Okay, and next we have post-translation, Lord, post-translational modification. And this is a covalent modification that happens after the protein has been made. It is often reversible and sometimes it is not reversible. It is the protein, the cell does, the cell changes this protein permanently and it just continues doing that function for the rest of its life. And the ones that are reversible, they often act as a switch so the cell can turn this on and off. Can somebody tell me why that might be? Why would the cell need to be able to turn these protein functions on and off?
So there's not like overproduction, and so
Yes, and remember that the cell is a dynamic place and it needs things to happen fast. And like you said, if there's too much of one thing happening, the cell needs to be able to stop that thing from continuing, or it needs to, or if it's not happening enough, the cell needs to be able to control it working. And one way the cell does this post-translational modification is through phosphorylation. It adds a phosphate group by a kinase or dephosphorylates and it removes a phosphate group by a phosphatase. The last, no, I think, is regulating protein function, nucleotide binding and hydrolysis. So enzymes that bind to ATP or GTP can hydrolyze the ATP to a ADP, GDP. We're recently learning about this in RAN GAP and RAN GDP
Right, GDP, right.
and RAN GTP. Yeah. ATPases and GTPases may use this activity to control other proteins function. We just talked about RAN and RAS, or they may be used as a molecular machines, using the energy and structural changes to power their activities. So like motor proteins, which we will soon learn about.
Yes. You wanna, okay. Okay. So yeah, this is the second question on our worksheet thing. So identify one atom with a positive dipole and one with a negative dipole. Why do they have these? This is similar to the first question on the mini midterm, if you guys remember that. And do you think this binding interaction has a low or high KD, and would mutating tyrosine to serine increase or decrease the KD? Yeah, take a couple minutes to do that. There'll be a lot of these questions that kind of combine multiple concepts in the course, which is what we're kind of trying to go for here. So yeah, take some time to work on that, and then we'll come back and talk about the answer. Also, that is supposed to say aspartic acid to alanine and not tyrosine to serine. that should be, I think, matched up with the worksheet now. Sorry, just one more thing to add. If you don't remember the amino acids, which you don't have to, you'll get the chart. Alanine is just one CH3 group as the R group. Okay, I think that's everything I wanted to say.
That's true. So that. Look. Myself that this is a question for previous exams.
This one's not.
It's not?
Mhm.
I thought it was.
The diagram is.
Oh.
The diagram's from the mini midterm, I think, actually. Is it? No.
This is from, I think, quiz 2.
Okay.
Quiz 1 or quiz 2? Quiz 2.
No, 4 and 5 are the ones that I just straight ripped from last year's exam.
Okay, cool.
I'm 3, I think I made up. Kind of.
You're good at
Based
questions.
on something else.
You're good at making answers.
Thanks. I've just taken a lot of NTA for a lot of lower classes. and that's it now.
This question definitely blew a lot of people off, but I'm in my zone. This didn't should have. But I just you want to answer your questions, not.
Yeah, I agree. That's why I wanted to throw it on because I feel like there's probably just some fundamental misunderstanding that's happening that's
Maybe. I'm
still
genuinely
talking. So it starts to quiet down, I'll call it. But people are still talking, so that's good.
You want to call it?
I don't know. What do you think? Maybe a few more minutes.
2 more. 35. Yes. I think if we give them more time, we can get that. Guys. Fingers crossed.
Yeah, it's kind of working.
Get them loosened up. How many classes have you seen from that?
Did 285 in the fall, 151 last spring, and then 285 now. But then I also took 285 in the fall with her in my sophomore year, and then I took a seminar with her in the spring. So this is my
The
fifth
285 seminar?
391. So this is my fifth work class.
And you're alive?
Yes. Well, I mean, like I didn't take the last 2, but I TA'd the last 2.
Still, you're alive?
Yeah.
It's definitely a lot more work than I assumed it would be.
Yeah, the TAing is a lot of work.
I was like, oh, as a student, it was a lot of work. So as a TA, it shouldn't be that bad.
And because it's a lot of work for them, it's a lot of work for us.
Boy, it was ironic.
Yeah. Yeah. Oh well, I still enjoy it.
I'm
Uh,
having
yeah.
fun, don't get me wrong. I
Yeah.
am having fun.
It is just sometimes the grading gets to you, especially the exam grading.
You're actually kind of insane for like getting this board done.
I just had time last night.
Wow.
I just had time last night.
Okay, that's actually crazy. You want to call it?
Yeah, sure. Do you want to do this one?
Oh, sure.
Cool. Turned it off. Sorry.
All right, what do we think? Okay, let's do the first part of the question first. What atom, any atom, does atom have a positive dipole and one with a negative dipole? Okay, let's shout out the positive dipole. Which atom are we thinking going?
NH3. NH3.
NH3. That's not an atom, but what atom is that one part of?
Um,
You can use the part. Trust me, you don't have to memorize any of this.
would be the hydrogen.
The positive dipole?
The positive dipole? Yeah. Okay.
In the NH3 group, the hydrogen would have the positive dipole.
Okay. And with that, the nitrogen will have the negative dipole. Um, okay, let's go. That's fine. Okay.
Electronegativity.
Okay. So how should I transition into electronegativity? What is creating these positive dipoles? How are these dipoles created in the first place? So we'll use NH3 as an example, right? We just said that nitrogen has a negative dipole and hydrogen has the positive dipole. Why?
Is it because the nitrogen is more electronegative than hydrogen, so it like pulls the electrons towards it, so there's an uneven distribution of electrons?
Exactly. So that's exactly what electronegativity is. And the dipoles are caused by the electronegativities and not the opposite. And as our lovely student over there just said, these dipoles are caused by the unequal sharing of electrons. Nitrogen is a lot more electronegative than hydrogen, so it pulls more of those electrons towards itself, and that gives it a partial negative charge, and giving hydrogen a partial positive charge. And this then allows it to interact with water, which also has dipoles as the oxygen is more. You guys know what water looks like, right? H2O. Okay. So the oxygen.
I'm trying to find one of my items.
What is it?
Sorry about it. I'm like actually gonna panic.
You're good.
Let's get an umbrella laying down here.
So oxygen has a larger electronegativity than hydrogen, and that causes it to pull the electrons to itself and give it a negative dipole, and that gives hydrogen a positive dipole. And because of these polarity there, water can interact with this NH3 group. Okay.
Can I ask, sorry, can I ask a quick question?
Sure.
Does anyone see an umbrella in here? That would be awesome if you guys could help me. I apologize.
What color is it?
It's like red.
Red umbrella? Anyone see a red umbrella near them?
This table and this table. Today.
No red umbrella?
Thank you guys.
That was fun. What about the next question? Do you think this bud interaction has a low effect?
Yeah.
Can we do the last two?
Huh?
Do you want me to do the last two?
Yeah.
Just go to mine.
Great. So for the second part of the question, do we think this binding interaction has a low or high KD? What is KD? What is KD? Can somebody shout it out?
The opposite of affinity.
Perfect. Yeah, it is the opposite of affinity. And KD is a symbol for the dissociation constant. So if you have a high KD, that means something is dissociating a lot, and you have a low affinity. You would have a low affinity if you had a high KD. So does this interaction have a high or low KD? Just like show of hands if you think it's high. Okay, show of hands if you think it's low. Looks like more. And yeah, that'd be correct. You can tell based if something has a high or low KD based on the number of bonds it's making and the type of bonds it's making. So this would have a low KD because it has good affinity for this binding pocket. You have ion interactions here or electrostatic interactions here, here. You have hydrogen bonding here, and then you have hydrogen bonding, or sorry, this is ion dipole. I didn't see those negatives. Ion dipole here, and ion dipole here, and then hydrogen bonding here. So that's a lot of favorable non-covalent interactions. And if we mutated aspartic acid or aspartate to alanine, would this increase or decrease the KD? Yeah. Yes. Yeah. This basically makes it have less affinity for the thing. So you would have a higher KD. They would dissociate more because that non-polar bond, it would be a CH3 here, would not be making as favorable interactions with this OH group. Perfect. So here's our answer. Take a minute to like look at that, write that down if you want to. Process it. Yeah, there's a lot of words there. And then I'll send out the answer key after this as well. So you'll get that too. Okay, now we'll move on to topic 3. So this is our protein sorting, so localization sequences, gene expression, and nuclear import and export, which I'm sure you guys all love very much. So yeah, we'll just talk about the central dogma first a little bit. So this is like Bio 151 stuff, but it's important to review. In the cell, you have DNA, which then gets transcribed into RNA, which is then translated into a protein. And that RNA is often mRNA, which is like mature RNA, which is moved to the cytoplasm and then translated by ribosomes. Yeah, all these proteins localize to different places in the cell based on what they need to do. They can go to the ER if they need to work in the ER. They can go to the nucleus if they need to do that, or they can stay in the cytosol. And so a lot of stuff we've talked about is the localization sequences that correspond to where these proteins are going. So a signal sequence here is something that indicates to the cell where to bring something. So it's a part of the amino acid sequence. So once the protein is started to be translated by a ribosome, something else in the cell can recognize this signal sequence and bring this protein where it needs to go. So if you have a protein that needs to stay in the cytosol, you'll have no signal sequence. It'll just stay there, doesn't need to be brought anywhere. And then there are specific signal sequences for the mitochondria, the chloroplast, the peroxisome, the endomembrane system. And if you guys remember, the endomembrane system is the ER, the Golgi, the lysosomes, and then eventually secretion outside the cell. And you can also have signal sequences for the nucleus. Yeah, so question?
Do you think no, like the typical sequences?
No.
No.
You'll be that, yeah, you don't need to memorize like each amino acid that's part of the sequence. I think most of what will be asked of you is just like, we have a question about this later too. If a protein has this signal sequence and this signal sequence, where will it go?
Okay.
Stuff like that. Yeah.
Not even like the hydrophobic, like the characteristics of like, oh, the hydrophobic.
That's helpful to know. You won't be explicitly asked, like, does this signal sequence, at least I hope not. In the past, I haven't seen it.
You would ask us to like elaborate on it though and explain it a different question.
It would be good to know, if anything.
Yeah, it doesn't hurt to know.
Yeah.
There's
Just in
a
case.
diagram. I don't know if we have it on these slides, or I think it's back here. Yeah. That shows the type of signal sequence that can be that corresponds to each type of organelle. But no, you don't need to know the amino acid sequences, and it's helpful to know whether or not they're hydrophobic or not. But I think the most important things to know about them is where they're located on the amino acid sequence. So N-terminus or C-terminus, right? Because you can remember something is translated from N-terminus to C-terminus. And if you have a signal sequence on your C-terminus, that's going to be recognized later than if you would have it on the N-terminus, stuff like that. So yeah.
I have a question.
Yes.
For the endomembrane system, since if it has a lysosome retention sequence, will show a lysosome. Why does it go to the lysosome with the lysosome retention sequence instead of secreting into like cell membrane? Because it has the cell membrane sequence while it's on the path, it stays in the cell membrane. How does the lysosome how does the lysosome protein get to the lysosome with its retention sequence?
I do not know that. Yeah, I don't know exactly how it's transported to the lysosomes. All you'll need to know for the scope of this class is that if it has a lysosome retention sequence, it's going to go there. And then just assume that cell membrane signal sequence is your like last step. Is that an okay answer? Okay. Yeah. So signal sequences we just talked about. I want to be on this slide. Yeah, so an ER localization sequence is required by all proteins going into the endomembrane system, which includes secreted proteins. These are located on the N-terminus, and importantly, these move into the ER, sorry, the protein moves into the ER while being translated. This is called co-translational transport. And this means that the protein is entering the ER in an unfolded state. Oh, yeah, sure. Yeah. What else did I want to say about this? I think that's you need your retention sequences, of course. So if you just have your ER localization sequence, your final pathway is being secreted outside the cell. So if you want to stay in the ER, you need an ER retention sequence. If you want to stay in the Golgi, you need a Golgi retention sequence. Stuff like that. For the mitochondria, these are located on the N-terminus. These also move into the organelle before fully folded. These kind of diffuse, they're grabbed by a protein and they diffuse through the cell in a half-folded state, upon which they're put into the mitochondria and they fold fully. And then once they fold fully in the mitochondria, they can't leave because the pores are too small in the mitochondria for a fully folded protein to leave. Yeah, any questions on any of those? You don't need to know peroxisomes. We don't care about those. Yeah. Good. Oh, yeah.
I just have a question, what's the difference between like a protein being like in the ER and like in the lumen phase?
There isn't one.
It's just.
The lumen is like the inside of the ER. So you have the membrane, you have the lumen. Yeah. Okay. Here's just some comparison here. I already talked about the ER trafficking and the mitochondrial trafficking. Nuclear import is a little bit different than these. That signal sequence isn't actually visible until the protein folds. So you have to fully transport or translate your protein in the cytosol before this nuclear import sequence is revealed. It's like if you look at the space filling model, for example, it'd be like a little patch on the outside. And so once this folds, it can be brought into the nucleus. And then, of course, you have nuclear export sequences as well, which can bring things out of the nucleus into the cytosol. The pores on the edge of the nucleus are pretty large, so it can fit fully folded proteins. Any questions on that? Yeah.
If Flores's transportation transition happened, then it's nuclear phase two, or like even though like they have other proteins.
Yeah. So the question was if Laura mentioned something like a translocation channel, is she also talking about the nuclear pores? So I assume you're talking about with like the ER, like co-translational transportation, like stuff like that. Yeah. Laura will just call it a nuclear pore if you're talking about going to the protein fully folded, and then your translocation channel is going to be where that protein is kind of translated while it's going into the ER. So now we get to nuclear import. Great. Yeah, so nuclear import begins in the cytosol when you need to move a cargo protein into the nucleus. This is recognized because that protein will have a nuclear localization sequence, which we just talked about. Where is that nuclear localization sequence? Is the protein folded or unfolded? folded, yeah. I heard like two people say that. So yeah, important will bind that nuclear localization signal and move through a pore in the nucleus into the nucleus. So that's this step right here. Once it goes through, it's in the inside of the nucleus, and you have RAN GDP. And RAN GDP has to exchange GDP for GTP using GEF. Is GEF an enzyme? No, it's not. Good. Yeah, GEF helps RAN exchanging GDP for GTP, just like GAP helps RAN exchange GTP for GDP, but they are not enzymes. RAN is the one with the GTPase activity. So after that happens, you have your RAN bound to GTP, and it will bind important. That causes the cargo to release, and you have important bound to RAN GTP, which then diffuses outside of the nucleus into the cytosol. In the cytosol, RAN hydrolyzes GTP with the help of GAP and changes shape to release important. So remember, all of this is happening because structure affects function. And if you have certain things binding, the structure of these proteins will change, causing the release. So if you have RAN GTP, this has good affinity for important, but then RAN GDP does not, which allows it to release important. I think that's everything with nuclear import. For questions on the exam for this stuff, you will definitely be asked, like, what happens if there's a mutation in this or a mutation in that? And we'll practice that in, I think, a slide or two. Before I move on to export, any questions on import? No. Okay, cool. Export time. So the cargo protein will have a nuclear export signal. Exportin binds RAN GTP with that cargo protein. Exportin binds RAN GTP, which allows it to associate with that cargo protein. And then they move through a nuclear pore into the cytosol. Once it's in the cytosol, RAN GTP exchanges GTP for GDP, causing it to release both exportin and the cargo, and exportin will cycle back into the nucleus. This one's a little bit less complicated. Yeah. Good with that? Cool. Here's GEFs and GAPs. We just talked about that. GEF stands for guanine nucleotide factor, and that helps exchange GDP for GTP. And GAP is a GTPase activating protein. And the GTPase hydrolyzes GTP for GDP. That basically means it's helping RAN exchange GTP for GDP. Yeah. As we just said, neither of these are enzymes. RAN is the actual enzyme. Yeah, I think that's good. No questions on that? Cool. All right, we'll get to our third question. This one's not on the worksheet. This is just like I think this is from your quiz, maybe. Yeah. Imagine a mutant cell where RAN-GEF can move between the nucleus and cytosol freely. What signal sequences would it need to be able to move around like this, and what would be its effect on nuclear import? Take like a minute or two to talk with the people around you, and then we'll review the answer. Yeah, sorry. That would be helpful, wouldn't it? Well, it's on the answer slide, so whatever. Yeah, so I realized I didn't actually give you a diagram, so you can't do that. So here's the answer. It would need both a nuclear import sequence and an export sequence if it were to be both in the cytosol and the nucleus. RAN-GEF in the cytosol would switch RAN to the GTP bound state, meaning there would be more RAN GTP in the cytosol, finding more important. So basically, you would have an accumulation of RAN GTP bound to important in the cytosol. That means there's just no important available to bind to cargo proteins and import them into the nucleus, which is bad. Yeah, sorry about that. I'll leave a diagram up for this one. Or this is not the right one. I have another question at the end. Okay. Yeah, so I talked about the central dogma of biology already. DNA is read by RNA polymerase. It makes mRNA. Ribosome reads that mRNA to know in what sequence the amino acids should be ordered. And the protein folds into a specific conformation due to the sequence of those amino acids. Yeah, so the cell wants to control all of this, right? You need regulatory things in the cell to have not make bad things happen. I don't know. Yeah, so you can control the rate of transcription by regulating the level of chromatin and these things called histones. I don't think we really talk about like methyl and acetyl groups. Basically, you can regulate how tightly DNA is wound around histones and whether or not the RNA polymerase can access that. General transcription factors also regulate transcription. So these help RNA polymerase bind to DNA, like Sigma 70, which we talk about a little bit. And yeah. Yes, and then there's also regulatory transcription factors, which can be positive and negative regulators, like with what we see on the trip operon. We'll get into this a bit later, but can anyone tell me if the trip repressor would be a positive or negative transcription regulator? You can just shout this out. It's in the name of the protein. Yes, good. It's a repressor, so it's negative. Yeah. Chromatin, again, histones react in DNA. Packaging determines which genes can be expressed. Chromatin has an open state in which RNA polymerase and transcription factors can bind to DNA, and they have a closed state. You don't need to know about euchromatin, heterochromatin. Those are just the names for those, so we didn't talk about that. Yeah, question?
The positive and negative.
Yeah.
For the negative regulatory proteins, do they bind to the operator of the promoter?
Yeah.
The operator region, and then the positive regulatory proteins, do they bind to the promoter? Like where do they bind?
Yeah. So it all depends on like what, I guess, in bacteria, which operon you're talking about, or in eukaryotes, like which DNA segment you're talking about. For the purposes of what we know, that repressor binds to an operator, which basically blocks RNA polymerase from coming on because that operator is right next to the promoter sequence. Sometimes you can have positive transcription factors that bind like on the other side of the DNA, like upstream of the promoter, that then like cause some sort of better affinity for that RNA polymerase to bind to the DNA. Yeah, is that
does it just change case by case?
Yeah, I would say so.
But the repressive proteins, they don't always bind to the operator, they do.
I don't want to say always because I don't know the scope of everything in regards to that, but for the mechanism we talk about, sure. Those negative regulatory transcription factors will bind to the operator. Yeah.
Right. But and do you need general transcription factor first before positive like transcription factor? Can you just have the positive?
That's a good question. I don't know. I would say you can probably have both.
But you don't like need the general.
I would say generals are probably more important than positive, because your positive is like just another upregulation, but your general is like what helps recruit that RNA polymerase to the promoter. Okay, cool. Yeah, so we talked about chromatin. RNA, after it's transcribed, needs to be processed and made into mature mRNA. I'll just go over this quickly because we don't really talk about this that much. I think it was just in one of the lecture videos. But you'll have a 5' GCAP, which is added to your RNA, and then a poly A tail. This protects the RNA from endonucleases in the cell, which will like, chew down the RNA and cleave it. Yeah, same thing with the poly A tail. It's just a bunch of A's on the end of mRNA that help it not be degraded. RNA has introns and exons. So introns are transcribed into that initial RNA transcript, but they're not translated. So once you have that initial RNA transcript and you're making your mRNA, those introns are spliced out with using something called the spliceosome. Exons are both transcribed and translated, so your exons end up in that final protein being translated into amino acids. And alternative splicing is where you can get like different exons in a protein. So you have like exon 1, exon 2, and exon 3 in one protein. And then maybe in another protein, it's the same exact transcript, but you only want exon 1 and exon 3. So it makes a completely different protein. And that helps increase the diversity of the proteins in the cell. Yeah. The rate of mRNA translation is controlled in the cell as well. That's controlled by ribosome binding to mRNA and the localization of the mRNA. So, we can localize that to the key bodies, like in RNA interference, and get it away from ribosomes, stuff like that. So basically, your mRNA is controlled in the cell, and like how long it's around and whether or not it'll be translated is controlled by the cell as well. That's an example of post-transcriptional regulation. Translational control is basically just the ability of the ribosome to bind that mRNA. Post-transcriptional control and translational control are kind of tricky to discern apart from each other. Laura's main thing is just anything that has to do with the ribosome actually accessing that mRNA is translational control. So post-translational control, we've kind of talked about already. So post-translational modifications like phosphorylation, dephosphorylation, that can change how the protein functions in the cell. Allostery can also change how the protein functions in the cell. And then your proteins can also be degraded. So they can be ubiquitinated, which is like a little tiny protein that gets attached to proteins. And then it tells the cell, bring this to the proteasome and break it apart. So that's an example of post-translational control. That's, the amino acid sequence of the protein is actually getting modified. Here's our diagram that we talk about. So we have our first step is when you are transcribing the DNA to RNA, you have transcriptional control. And you have RNA processing control, which is like splicing, which you then get to your mRNA. That mRNA is transported out of the cell, or out of the nucleus, sorry. That mRNA can be degraded or it can be translated into a protein. And then that protein can be degraded as well, or the activity of that protein can be controlled. Any questions on gene expression? All right. Okay, here is our question on the worksheet. A protein is engineered to have an ER localization sequence on the N-terminus, a nuclear localization sequence, and a Golgi retention sequence. Where would this protein be found? Take 2 minutes and talk about it with people around you. Okay, does anyone want to tell me where this would end up? I'm not flipping to the answer like last time, so someone's actually got to raise their hand. Sure.
That protein would end up in the Golgi because the RNA localization in the N-terminus, which means co-translocation translation would occur. The SRP would be bound to the I think it's called the signal something protein.
Yeah.
It would bind to the IR localization signal, take it through the endomembrane system. The nuclear localization signal would be useless there, so only the Golgi retention signal would matter, and then it would stay in the Golgi retention, the Golgi loop. I was just called Golgi inside.
Just Golgi, yeah. Perfect. Yeah, that was a perfect answer. So because your ER localization sequence is on the N terminus, that's going to get picked up by SRP, which will bring that amino acid and ribosome to the ER. It'll be co-translationally transported into the ER, where that protein then folds. That nuclear localization sequence is only seen once the protein folds. So when it's in the ER, it can't move out. That nuclear localization sequence means nothing because it's trapped in the ER. And then it'll continue going through the endomembrane system until it reaches the Golgi because it has that retention sequence. So perfect. Great job. Cool. Now, yeah, here's the diagram. Now you remove the ER and Golgi signals from the protein, making it only have a nuclear localization sequence. However, your cell has a mutated version of GEF that cannot interact with RAN. What would happen to this cargo protein? You can think a little longer with this one. This one, you have to look at the diagram. Going kind of quiet. Does anyone want to try and take a stab at this, or do you just want me to explain it? See anybody raising their hands? So, oh, you want to go again? Sure.
The cargo protein would likely be stuck in the nucleus with the importance still attached, since without the RAN gap to turn the GDP into the GDP into the GTP, the GTP would then not interact with the important, so the important wouldn't be changed in its conformation state to release the cargo protein.
Yeah, perfect answer again. So once that it says it has a nuclear localization sequence, it will be bound by important and brought into the nucleus. However, since GEF exchanges GDP for GTP and helps RAN that, you won't have any RAN GTP in the nucleus, and so it can't bind important to change its conformation and release that cargo. So you would just have an accumulation of important bound cargo in the nucleus that can't really do anything. And for the little bonus question, GDP to GTP interaction with RAN is an example of protein activity control, that nucleotide binding and hydrolysis we talked about earlier. Questions?
I remember she said in class that RAN can do some stuff without GAP and GAP. Is this one the thing that would just take a really long time, or can it not release the cargo protein of it?
Yeah, so the question was, there's some instances where RAN can like have activity without GEF or GAP. And would this be one of those instances? I would say no. RAN's basal activity is a GTP ACE, so it'll be really slow at exchanging that GTP for GDP, which you see with GAP in the cytosol, but not in the nucleus. Here, you actually have to have that GDP dissociate and then bind to GTP. So its intrinsic activity is like GTP ACE activity. I don't know for sure. It could probably dissociate eventually, but again, it would take a really long time. So for the purposes of this class, we'll just assume without GAP, it's stuck in GDP bound in the nucleus? Good question though. Okay, cool. Now we're gonna move on to, uh-oh. Let's confuse what I. Well, hopefully it doesn't die while it's recording. Yeah, now we'll move into gene expression regulation. So that is transcriptional regulation in prokaryotes. So this is in eukaryotes, you have your separation of the nucleus and the cytosol. You have ribosomes and RNA polymerase in different time and in different areas of the cell. In prokaryotes, this happens at the same time because there is no nucleus. So you can have something being transcribed as it's being translated as it's being transcribed. So you can have both RNA polymerase and ribosome bound to the same DNA, RNA complex type thing. Again, we talk about basal and general transcription factors. So this is not like promoter specific stuff, but stuff that helps recruit RNA polymerase to the promoter.